To inform you that during the break, we will have the opportunity to hear from Travis Hardcastle from Horizon Discovery, who is sponsoring this symposium. I would like also to kindly ask you to ask your question through the QA box, and I will ask the question for you. So Wallace, if you are ready, we are looking forward to your talk. Wallace, you have to unmute yourself. Wallace, unmute yourself. Yep.
Okay, am I unmuted now? Sorry about that.
Yep.
I was so excited to share my screen that I forgot to unmute myself, and then I couldn't do it. Okay. Can you see me and hear me?
Perfect.
Okay, good. So it's important for all organelles to control their size, and so I think there's a general question of how organelles do that. And what I'm going to tell you about today is our study of one particular organelle that we think is very tractable for studying size control questions. And that's the eukaryotic flagellum. So this is a microtubule-based structure composed of 9 microtubule doublets that push out a protrusion of membrane. And it's really the control of the length of those microtubules that we mean by length control in this organelle. So the question becomes, how does it control the size of the structure, the length of the structure? And we're doing all of our work in Cleminomonas, which is a green alga with genetics similar to yeast. So first, the reason why we even think there is such a thing as length control is that if you remove the flagella from a cell, they'll grow back to the length that they were at before you removed them, and they do so with decelerating kinetics. So the rate of elongation is about 1 over the length. So it appears that they know how long they should be, and they asymptotically approach that desired length. So the question becomes, how do they do this? How do they know how long they are and how long they should be? What is the control system? What we know is that when a flagellum first grows, here I'm showing microtubules in green, they grow by assembly of tubulin out of the distal end. And what we found later on was that once the flagellum has reached its final steady state length, it's still assembling new tubulin at the end. And the reason why it stops growing is that that assembly is being balanced by constant removal or disassembly of tubulin also from the distal end. So there's this constant balance of assembly and disassembly. And we also know that assembly requires an active transport called intraflagellar transport. And the reason why it needs that is because the protein is made in the cell body and has to be transported out to the tip where the assembly is taking place. So what is this transport? Intraflagellar transport is a kinesin-driven motility where you have a kinesin that grabs a particle called an IFT particle that's covered in tubulin and other binding sites. So it can grab cargo and carry it out to the tip. These particles aggregate into arrays called trains, and the whole train moves out to the tip, powered by kinesin, drops off the cargo, which includes tubulin and other proteins, then the whole train goes back for more. So the picture we have is that we have assembly and disassembly constantly going on. Assembly requires transport. And if you wanted to control size in a system like this, whatever the control system is, it has to be able to make sure that the two rates of assembly and disassembly balance each other if and only if you're at the correct length. So how does that happen? Well, we know that the rate of disassembly is apparently length independent. So tubulin is being removed at the constant rate regardless of how long the flagellum is. And we also know that so therefore assembly has to be what's controlled. And we know that assembly depends on IFT, on intraflagellar transport. If we tune transport up and down using mutations, we see a concomitant change in the assembly rate. So that raises the question, maybe the transport itself is what's being regulated as a function of length. So the question is, what is being regulated? And we know that IFT is very precessive. Once a train with its cargo enters the flagellum, it will move to the tip. So the only way you can really control this is at the level of injection, at the level of entry of those trains into the flagellum. So what regulates that? So over the years, we've done a lot of quantitative imaging using TURF and other methods. And so we know a lot about how the amount of IFT going in varies over time, for example. And the main take-home message is that the rate at which IFT trains are being injected into the flagellum, IFT particles is going as 1 over the length. So longer flagella have a slower rate of injection. So we get the following model where I'm plotting rates versus length. In red, I show the rate of disassembly, which we said is independent of length, so it's a flat line. The rate of assembly is presumably being determined by the rate of transport and therefore IFT injection. So this should scale as 1 over the length. So that gives you two curves intersect only at one value of length, and that's a steady state solution, and you can convince yourself that it's actually a stable solution. So this model can explain a lot of phenomenology that was previously known about this system can be accounted for. For example, that decelerating kinetics of regeneration that I showed you earlier on. So at some level, it can explain everything that we know. On the other hand, it doesn't actually explain anything because we've just changed the problem of what determines this 1 over L dependence of IFT injection. So we imagine that there's some kind of a pore or mechanism or gate at the base of the flagellum. People have argued that there's actually a nuclear pore-like structure there that's determining IFT being able to enter the flagellum. And whatever that mechanism is, it's somehow dependent on the length of the flagellum, which requires some kind of a length sensor that's then able to control injection at the base. So the question becomes then, how would a cell measure length of a flagellum? And so this has become a major focus of our group, is trying to propose models for how the cell might measure the length of the flagellum and then test those models. So one of the earliest models that we had, and I'm going to run through two of them that we've already ruled out. So one of the earliest models we had is that there was a fixed pool of IFT trains circulating back and forth and not actually truly injecting into the flagellum or leaving. And this is the original model that I had proposed back when I was a postdoc. This has now been ruled out by a number of experiments in our lab, and this is just one paper in which we've used fluorescence recovery after photobleaching to show that, in fact, there's rapid exchange of IFT with the cell body. So we can rule out this sort of fixed pool model. Another model that we really liked was this idea of a time of flight sensor, where you put a clock on the IFT particle, send it out, bring it back, and read off the time elapsed on the clock, and that would give you a measure of how long the flagellum is. And there's various ways to build a clock using molecules, but we've been able to rule out this entire class of models by slowing down the rate of return of the trains and showing that you don't get a consistent effect on injection. So this was published by my postdoc Hiro Ishikawa in 2017. So we've ruled out, we think, these two models, both of which I kind of like because they're elegant. So now we're interested in a third model proposed by Joel Rosenbaum, based on electrophysiology studies done by Beck and Uhl, who had shown that, in fact, in the Cleminomonas flagellum, there are calcium channels, and that the density of those channels is independent of length. So longer flagella would have more channels, and therefore would have a higher, the flagellum as a whole would have a higher conductivity. So you could have a higher current of calcium flowing in through the flagellum as a whole. And so Joel's model was that if this calcium is somehow able to inhibit IFT injection, then longer flagella would have more calcium flowing in, that would shut down IFT injection, and you would see this fall off of injection rates as the length increases. So that was the model. It was very abstract when he first proposed it because there was no mechanism proposed for how calcium would regulate IFT. That's now been provided by Junmin Pang's lab, who has shown that one of the kinesin motors of IFT FLA8 can be inhibited by phosphorylation by a calcium-dependent protein kinase. And when it's phosphorylated, the IFT motor no longer is able to inject into the flagellum. So the model is that higher levels of calcium would trigger this kinase, and that it would phosphorylate the kinesin and prevent it from being injected. So the predictions of this model are two. One, it should be the case that as flagella get longer, there is actually more calcium coming in. And second, if you could somehow reduce the calcium entry experimentally, you should actually see an increase in IFT injection because you've removed this inhibition that the Pang group has shown, and you should therefore see faster growth of the flagella. So to test these ideas, we've worked with collaborators to construct Cleminomonas strains that contain calcium biosensors. So this is what's shown on the right panel. And so if you watch these movies, you can see these little flashes of calcium signaling. And based on quantifying images like this, of course, now I can't, okay. What we see is that indeed, as flagella get longer, the amount of calcium going into the flagella during these flashes is proportionally larger. And also, we can look at a mutant called PPR2, which is defective in the calcium channel of the flagellar membrane. And we see that there is still calcium getting in by other means, but you see a reduction in the amount of calcium at any given length. So using that mutant, you can then ask what happens to injection if you reduce calcium entry. And the prediction was that if you reduce calcium, you should alleviate the inhibition of the kinesin and therefore see more injection. But in fact, we see the opposite. So the controls, wild type cells shown in blue, we see again, as the length increases, injection goes down. Shown in red is the PPR2 mutant, and we see there that at any given length, you actually see less injection than a wild type, not more as the model would have predicted. And we see that same effect if we chelate calcium EGTA. You see less injection rather than more as the model predicted. Also, if we look at growth, the prediction was that without calcium, you should have more injection and therefore faster growth, and we see the opposite. So in the PPR2 mutant, we see slower growing flagella. This is length versus time, so you see a slower growth rate. So going back to this really quite elegant calcium-based model, we find that the first prediction is achieved, that is, calcium entry does appear to be length dependent, so longer flagella have more calcium going in. But the second set of predictions that reduce calcium entry should increase IFT and lead to faster growth, that is actually not actually seen. And so we think we can rule this model out. So now we're considering a fourth class of models based on diffusion. And this is based on the fact that the kinesin motor that drives IFT moves processively by its own activity out to the tip, but after that, it pops off of the flagellar trains and returns by diffusion. And this has been directly demonstrated by Ahmed Ildiz at UC Berkeley, who has actually measured the diffusion constant of kinesin. And so the idea is that the kinesin has to diffuse all the way back, and you can't have more trains coming in until enough kinesin has diffused back to have another injection event. And so we've done a number, so Nat Hendel and my lab has made a number of models, agent-based and analytical, to confirm that indeed this kind of model can explain the phenomena that we see. So that as the flagellar length is shown in the middle graph below, as the flagellar length increases, the injection rate does in fact go down. And as shown on the right, you do get this decelerating kinetics of growth. And so now we're at the stage of trying to figure out how to test this model. And if anyone in this esteemed audience has a good idea for how to test such a model by, for example, tuning the diffusion constant, I would love to hear it, because it's very difficult to change the diffusion constant of a protein. So I want to close by thanking the people involved in the group. This is our length control team. Also, we have many collaborators that have helped us, and I'm grateful for our funding, and I'm grateful for the organizers for giving me the chance to speak, and I'll stop and take any questions.
Thanks a lot, Wallace. If there is anyone who has a question, please go to the QA. We have already one question from Nevan Krogan. The pore at the base, do they have the same pore proteins that are at the nuclear pore?
It's a great question. This is actually really controversial. So there have been reports that using antibodies and so on, that there are some nuclear pore complexes there. There's very good evidence from Kristen Verhees' group that the RAN system is actually involved somehow in transport into the flagellum. So that part does seem to be similar. Whether there's actually nuclear pores there is really less obvious. You certainly see holes in cryo-EM, so there's something like a nuclear pore. We haven't had good luck ourselves with antibodies for a nuclear porin, so we're not really sure if it's really, the same exact molecules, but it's certainly something very similar, if not the same.
Okay, thanks a lot. We have another question from Marie-Hélène Verlac. How do you coordinate plasma membrane growth with microtubule growth?
So our current thinking is extremely simplistic. We just imagine there's surface tension and you push the microtubules out and they just drag membrane along. It's obviously not going to be that simple. We know there's secretion of vesicles at the base of the flagellum. So what coordinates all that? I think is a fantastic question. I know absolutely nothing that speaks to that really. And so, we tried for a while. What I'd love to be able to do is to pull tethers off of the main plasma membrane and ask, if you pull tethers off, does that force the flagellum to shorten? The problem is there's a cell wall which makes it impossible to pull the tether. And so we may need to go to a different system to actually ask that. I think it's a very interesting question that's totally unexplored, really.
We have another question from Bill Sullivan. Does the pore at the base change size with length?
I do not know of any data to that effect. So people have imaged the pores in Tetrahymena, especially, have really, really beautiful images, but I don't know of any studies that have actually done that as a function of length.
Okay. Another question from Bruno Goud. Is there any regulation at the transcriptional level?
Yes, so when flagella are growing, genes encoding basically all flagellar proteins are upregulated many-fold. So there's clearly transcriptional control. You don't need that control to achieve a fixed length. So if you poison transcription and translation and then chop flagella off, they'll grow back to a defined length. It's shorter than normal because they can't make enough protein, that they're relying on their pre-synthesized pool. But other aspects of length control are fine, so that they will actually stably maintain that partial length. So we think that transcription is definitely involved in determining what the final length is. And this big question now is, well, how do those genes know how to turn on? And the general thinking has always been that there must be an active signal that the cell determines the loss of the flagellum and triggers transcription of these genes. We're thinking now that may be quite different, and work from a former lab member, Ivan Zamora, and my current student, Karina Perlasa, suggests that in fact, the genes are being inhibited by some inhibitor, which is then sequestered in the flagellum. So as it grows faster and faster and faster, there's less inhibitor around in the cytoplasm, and that allows the genes to turn on. And then as length equilibrates, the repressor can build up again and turn the genes off.
Marie-Hélène, maybe you can start sharing your screen. She will talk about cytoplasmic remodeling compartmentalize the oocyte nucleus.
Okay, do you hear me?
Yes, perfect.
Okay, so thanks for the invitation. So indeed, today I would like to share with you some work where we would try to convince you that forces from the cytoplasm can actually compartmentalize the oocyte nucleus. So in many cell types, the location of the nucleus is very important and instructive for the cell. And this is typically the case of human and mouse oocytes, which position their nucleus in their center at the end of their growth. And a failure to do so correlates with a failure to achieve further meiotic division and further embryonic development. So we have shown that central nucleus positioning in mouse oocyte requires the presence of an active cytoplasmic mesh that is nucleated by forming a straight F-actin nucleator. And not only does this cytoplasmic mesh promote nucleus centering, but it also puts the whole cytoplasm in motion, so mixing all the cytoplasm, and it imposes forces on the nuclear envelope, promoting nuclear envelope fluctuations that in turn mobilize chromatin and regulate about 200 genes. So this work has been published. So in this work, we actually addressed when does exactly this cytoplasmic activity that is mostly due to actin filament arises during oocyte growth. So while at the end of oocyte growth, so the cell tends to increase its volume, and we observe that at these stages, the density of actin filaments tends to increase while the nucleus becomes centralized. And at the same time, we could correlate this increase in cytoplasmic actin density with the increase in cytoplasmic activity. During these stages, the nucleus volume stays constant. Importantly, during these late stages of oocyte growth, the chromatin undergoes a major remodeling. That is, from a decondensed chromatin around 2 nucleoli, it will end up as a condensed chromatin around a single nucleolus. So, yeah, as I said, the 2 nucleoli will fuse into a single nucleolus that is roughly twice the volume of this single nucleoli. So nucleoli are nuclear bodies, that is membraneless organelles that are inside the oocyte nucleus, and there are many types of membraneless organelles. They are condensate of protein and RNA, and they have a major role in transcription, but also in mRNA processing. So we performed an extensive screen for all endogenous nuclear bodies. I'm only going to show you 2. Chiral bodies and splicing speckles at these late stages of oocyte growth that we are interested in. So we observe, for example, that chiral bodies tend to appear at mid stages. So you see we have multiple chiral bodies, and that they will tend to become bigger. We also observe that splicing speckles at earlier stages are also very numerous and tiny, and they will progressively become less numerous and bigger. In oocyte coming from the forming 2 knockout strain that don't have a cytoplasmic mesh, that have very low cytoplasmic activity and very low nuclear envelope fluctuation, we observe that chiral bodies are more numerous and tinier, smaller than in control, and splicing speckles are also more numerous and smaller than in control oocytes. So we decided to follow in live the behavior of splicing speckles, first because they were present at all the stages we are interested in, and also because they are quite numerous, so easier to follow in this volume of the oocyte nucleus that is 30 micron wide, so big nucleus. So splicing speckles, they behave like dynamic liquid-like droplet able to fuse or coalesce, and you see an example here. So this is with an appropriate marker. And so we followed these splicing speckles at all the stages of growth we are interested in, and we measure their diffusion coefficient, and we could observe that there is a correlation between the droplet diffusion, so their diffusion coefficient, and the cytoplasmic activity. When the cytoplasmic activity at the end of oocyte growth, the diffusion increases. So we wondered, is it only a correlation, or can we play with cytoplasmic activity and see whether we have an impact on droplet organization? So, for example, in a fully grown oocyte, we can follow splicing speckles droplet, and we can treat these oocytes, or these nuclei, with cytochalasin D, a drug that will depolymerize actin, and follow the behavior of these droplets. And you see, I think you can appreciate that they become less mobile and tinier. So we did this in a systematic manner using either drugs or genetic tools to either increase or decrease cytoplasmic forces. So in our model system, the mouse oocyte microtubule actually have a negative effect on cytoplasmic activity. And when we depolymerize them, we tend to increase cytoplasmic activity, so increase the forces from the cytoplasm, increase the nuclear envelope fluctuation. And in this condition, we observe an increased diffusion of our droplet. On the contrary, when we decrease cytoplasmic activity, sorry, it's my cat. When we decrease cytoplasmic activity in all these conditions, we tend to decrease the droplet diffusion. So similarly, when we looked at droplet size, we saw that when we increase the cytoplasmic activity with nocodazole, a drug that targets microtubule, we observe an increase in droplet size, while conversely, when we decrease the forces from the cytoplasm, we observe a decrease in droplet size. So this suggested that there is a correlation between the activity and the droplet size and their capacity to fuse. So we decided to challenge this hypothesis. So the hypothesis that cytoplasmic activity has an impact on the diffusion of droplet or their coalescence by using 3D agent-based simulation. So in this simulation, we have simulated droplets in orange and the chromatin only here we represent the nucleolus in gray. And so we could, in this simulation, both simulate chromatin condensation and droplet behavior, and so their diffusion. And we could test different range of cytoplasmic activity. And this simulation showed that actually there is a good correlation between the prediction from our simulation and the behavior of droplets that we could measure experimentally, arguing that the hypothesis that the cytoplasmic activity influences droplet diffusion is relatively possible. Importantly, we could also measure the contribution of chromatin condensation, and we could see that actually, indeed, while chromatin condensation has an impact on the capacity and the kinetics of droplet coalescence, the activity is mostly the major player involved in the kinetics of droplet coalescence in this model system. So what are the consequences of an impaired nuclear body organization at the end of the oocyte growth? So we compared the transcriptome of control or formin 2 knockout oocytes, which have very low cytoplasmic activity on off-center nuclei. So we observed that actually we had very strong impact on exon usage. So many exons were overrepresented and many were underrepresented. This corresponded to approximately 3,000 alternative splicing events that were distributed all along the process of mRNA splicing, arguing that the quality of the oocyte transcriptome was affected, and probably the splicing of messenger RNA and of many RNA. Also, what we wanted to test was the conservation during evolution of this capacity of the cytoplasmic activity to impact nuclear organization. So we did that in collaboration with Soline Chanet and Jean-René Wien. And so we tested in the Drosophila oocyte, which also harbors cytoplasmic steering of its cytoplasm at the end of its growth. And we use various mutants to either enhance cytoplasmic activity or, on the contrary, decrease cytoplasmic activity and observe the impact on splicing speckles. So an increase in cytoplasmic activity induced bigger splicing droplets, but less numerous, and on the contrary, a decreased cytoplasmic activity induced more numerous but smaller droplets. So we would like to conclude that the cytoplasmic remodeling that we observe at the end of oocyte growth improved not only the amount, but also the quality of maternal messenger RNAs that are present in the oocyte at the end of its growth. And we know that these stores are essential to support further oocyte development, because at that stage, transcription is very low and will be reactivated again only later on. So in a way, this cytoplasmic remodeling that compartmentalizes the oocyte nucleus at the end of growth improve female gamete fitness. And so I would like to acknowledge the people who did the work, so two extremely talented postdocs, Adèle Aljord and Gaël Le Thor, and all our collaborators. And thank you for your attention.
Many thanks, Marie-Hélène. We have a first question from Nevan Krogan. Overexpression of FMN2, do you increase droplet diffusion?
So we can actually rescue the fluctuation of the nuclear envelope. So we rescue all the aspects of nuclear positioning, nuclear envelope fluctuation that are defective in the forming 2 knockout oocytes. But we didn't test whether we specifically this experiment, because it's a kind of difficult experiment to do, because forming 2 is a very long protein, and so we inject cRNA, and so we didn't do this specifically. But we rescue all of the features, and we actually rescue the rest of the division. So probably, yeah.
Thanks. We have another question from Mehdi Bouhadou. Do you think there may be therapeutic implication of this work in potentially trying to alter cytoplasmic activity in order to affect nuclear events, transcription, etc., to treat disease?
So indeed, we think that maybe this phenomenon that we are revealing in the mouse oocyte might be at play in other types of cells, like, for example, cancer cells, which have been shown to have increased cytoplasmic activity. And so maybe, you know, by using the cytoplasmic activity is a very rapid manner to actually modulate your transcriptome, because it will affect splicing. As we see in our, it seems to be associated to correlate with altered splicing, and so it's a very rapid way, you know, to modulate the expression of your genes. So indeed, maybe in cancer, but also maybe also in ALS, you know, this neuropathy that affect motor neurons that are also very big cells, and it might also be at play there.
Okay, perfect. Thanks a lot, Marie-Hélène. We will have now the next speaker. I think there is no other question. No, there is no other question. The next speaker is Olivier Espedy from Collège de France. Olivier, can you maybe share your presentation? Perfect. So he will talk about the adaptation of E. coli pathobionts to life within macrophages. The stage is yours. Olivier, you have to turn on your mic. Yep.
Okay. Hello. Is it okay for the screen?
You have to put it in presentation mode.
Here. Come on. Yeah. Okay, so thank you for the invitation. So today I will present our recent work on the adaptation of an E. coli pathobiont to the life within macrophage. So the bacteria we are interested in is this family of adherent invasive E. coli. They have been found in patients with Crohn's disease, and actually they benefit from the dysbiosis that these patients present to colonize the gastrointestinal tract. Nonetheless, they colonize the lumen, but they also adhere to epithelial cells, and they can invade them. So they are called pathobionts rather than pathogen because yet no disease have been discovered involving this bacteria in unpredisposed individuals. The step we are interested in is the next one. It has been observed in biopsies that IEC can be found in macrophages from the lamina propria. And in these macrophages, they can multiply, and they multiply in phagolysosome, so toxic vacuoles that is dedicated to destruction of bacteria and other organoids. So our interest here is how those IEC manage to deal with multi-stresses without detoxifying the vacuole. So just as a piece of introduction, let me just precise that bacteria are very well adapted to deal with stresses and deal with changing environment. As soon as the stress hits the bacteria, protein, lipid, or DNA can be modified. This will signal to transcription cascade that allow gene expression of detoxifying enzyme, efflux pump, etc. If these systems are functional enough, bacteria grow again. If not, they may change their growth behavior, their morphology, and eventually they can also change behavior or single cell behavior or eventually collective behavior, such as predation and sporulation and things like that. So to address this question about survival of IEC inside macrophage, we ran the first series of assays, and the first one was combining RNA-seq and dual RNA-seq. So in macrophage infected with IEC, we check that they are growing in toxic vacuole. This is illustrated here by the fusion with the with lysosome marked by LAMP1 protein. At any time of the kinetics, we compare this dual RNA-seq experiment with the RNA-seq performed in culture, regular culture or stress culture. So to make a long story short, we can summarize this result on this PCA analysis. Basically, IEC inside macrophage are stressed and they adapt their growth to the macrophage condition. And I will come back to that later. So now when you look at the single cell level, so this is microfluidic analysis of infection, we see that it's much more complex. Some IEC are able to kill macrophage. This is what's happening here. You have IEC in red and the macrophage will die. Some are able to limit the infection, and some are not able to limit the infection, and the infection propagates, the colonization propagates. So this quantitative analysis of this kind of way gives rise to these noisy curves. But the main phenotype we can observe is the one illustrated by macrophage 9 here. It's a long lag phase and an abrupt multiplication step. So an abrupt increase of number of bacteria inside the macrophage at some point of the infection. If we go back to population analysis, counting viable bacteria now, we can draw approximately the same type of curve, a lag phase, an abrupt peak where the population of bacteria dramatically increase, and a slow decrease as macrophages are clearing bacteria. This is very different from what we observe with commensal bacteria that can be eliminated quite fast by the macrophage. Nevertheless, we can observe a lag phase that is almost the same as the pathovar strain. Another thing that we observe is the appearing of these large vacuoles containing many bacteria. And this is illustrated here. So it's synchronized with the peak. And these large vacuoles resist to the macrophage for a long time. And at the end of an infection, let's say it's about 5 days of post-infection, we can still observe these vacuoles while most of the single bacteria have been cleared here. So we have a kinetics in 3 steps: a lag phase, post-infection, that stay for 7 to 10 hours, a replicating phase, and a persistent phase where only the big vacuoles with a large number of bacteria can persist. So in the last slides, I will focus on this transition. What's determinant make bacteria going from lag phase to replicating? So one first element that we observe is that there is a huge heterogeneity in the stress response in these bacteria. So this is an acidic stress reporter, but we test many different biosensors. And you can see that 6 hours post-infection here, you can observe, even in the single macrophage, very different stress responses. Later, this difference is still present, but seems to segregate according to the vacuole the bacteria are in the different macrophages. To this difference in stress responses, we can superpose a difference in the growth capacity of the bacteria. So the bacteria we are infecting the macrophage with are replicative bacteria. If they stay replicative, they have nearly 100% of chance to die immediately post-infection. However, we noticed that many of them switch to a non-replicative state where they can tolerate the stress. And a small proportion of these non-replicative bacteria can even tolerate a huge amount of antibiotics. They are totally tolerant to antibiotics, but they are not resistant, just tolerant, not mutant. So, for example, how we distinguish that is with an FTSZ GFP marker. So the FTSZ is a tubulin-like protein that marks the division ring. And you can see that for our post-infection, most of the bacteria don't present FTSZ ring. If you mutate these stress responses, you can see that these bacteria get to divide, but their survival is severely affected. Another aspect that we discovered is that among the few bacteria that are able to make colonies, they need to secrete an exopolysaccharide matrix. So this matrix is similar as the one you can find in biofilms. You can observe it. This is a STED super resolution microscopy with a WGA labeling inside the vacuole around each bacteria. So this matrix may protect the bacteria, and mutants affecting the production of this matrix are clearly affected in the number of large vacuole with a large number of bacteria, and survival here again is affected. So lastly, we observed that one of the important players in this stochastic behavior is the induction of a pathogenicity island shared by many different bacteria, such as Yersinia pestis. This island is able to produce a system that capture iron from the host, and only the bacteria that are able to induce it, as you can see on this montage, 6 to 8 hours post-infection, induce the pathogenicity island and start to make a replicative foci, while single bacteria are still red and they don't present this induction. So altogether, these results, we really pay really attention to quantitatively analyze them, and with Sylvia Demonte and Anna Cheng, we were able to propose models, mathematical models explaining the growth property of these bacteria inside macrophages. So I don't go into the detail of the model, but this is published, but we can now use this model to create virtual mutants. Virtual mutant of the bacteria, but also virtual mutants of the macrophage or drugs similar. And we can model the behavior of virtual bacteria during infection and propose new experiments. So I finish here just by showing you this movie that illustrates what we think these communities of bacteria are and how they can contribute to long-term infection. Actually, we think that rather than a short-term protection, they might be more like a long-term protection. Here you can see a dying macrophage that will release these two communities, and they will be immediately catched by another healthier macrophage. That's what you see. Here, with this system, you propagate the niche, you propagate the load of bacteria, and it can be seen as an ecological niche because we can see that with different bacteria, and in this ecological niche, they may exchange goods and genes and promote evolution. So I end up here. This is quite old picture. You can still see Notre Dame in good shape, but this is to show that most of this work has been done by Gaël Demar and Victoria Prudhomme, who left my lab. Actually, Victoria is now in Suzanne Noble lab in San Francisco in UCSF. So feel free to go to talk to her if you are around. And thank you very much.
Thanks a lot, Olivier. It was very, very, very clear. I just remind everyone that you can ask questions through the QA box. We have a first question from Nevan Krogan. Has a genetic screen been done in macrophages to identify host genes involved?
Yes, so these bacteria have been discovered. IEC is a family of bacteria, but not a strain. So there many diverse E. coli bacteria can be IEC, can invade macrophage, and this is under study by a number of labs that are interested in Crohn's disease. In our essay, what we did is an RNA-seq analysis to analyze the response of the macrophage. The data suggests that the macrophages are very aggressive toward the bacteria, and that the bacteria can interfere with iron homeostasis of the macrophage. So maybe there is a hint here where we can go to fight or see what's happening in model organisms with the macrophage.
Thanks a lot. Yeah, Nevan is saying that there is maybe a collaboration on this possible. Yeah, because you talk a lot about iron or obviously, so you do believe that the idea of catching iron to have chelator for iron would be good to remove this infection. Is it right? Do you understand this right?
Yeah, well, it might be more complex than that. So limiting iron, bacteria really need to catch iron, and limiting iron accessibility is a common defense of infected hosts. Lactoferrin is at play for that, and extra. The problem in the specific case of Crohn's disease is that patients have already a lack of iron due to bleeding and etc. So this might be a challenge to go on this particular aspect for treating patients with Crohn's disease. One thing I didn't mention here, and you have to keep it in mind. We don't think the bacteria is a cause of the disease. The bacteria is a passenger of the disease and might just increase some of the phenotypes, such as some of the etiologies, such as the inflammation. So effort to eradicate this bacteria might not be the main target, and is not clearly the main target on Crohn's disease strategy.
Okay, thanks a lot, Olivier. I think there is no other question for you for the moment. So okay. I suggest that thanks a lot. So I suggest that
we go very much.
We suggest to go to the next speaker, which is Maria Teresa Teixeira. She's working at the Institut de Biologie Physico-Chimique à Paris. And she will talk about how telomeres cancel poly. Maria Teresa, can you maybe try to share your screen?
Hello, everybody. I think it's okay. Am I sharing? Do you see something?
Yes, perfect.
Okay. So hello, everybody, and thank you so much for giving me this opportunity to share our work with you, because it's a nice example of a collaborative work involving different fields and of quantitative biology. So our lab focuses on telomeres, who were first described by Liz Blackburn, who I don't think believes any, believes any presentations at UCSF. So telomeres are the ends of chromosomes in eukaryotes, and they are composed of repeated sequences, and their main function is to maintain chromosome integrity by preventing fusions and degradations. But telomeres display a highly dynamic structure, and in fact, they shorten with cell divisions. And this is compensated by telomerase, a reverse transcriptase, resulting in chromosomes having a variable number of telomeric repeats at their ends. However, telomerase isn't expressed in somatic cells of many multicellular organisms like humans, and this leads to extreme shortening and replicative senescence. At a molecular level, a replicative senescence corresponds to the activation of the DNA damage checkpoint. So telomeres are recognized as DNA damage and cell cycle arrests. And in most cancer cells have bypassed this step, but this transition is not fully understood. So one can ask, what is the robustness of replicative senescence state, and why is it infrequent but still possible to bypass? In budding yeast, telomerase inactivation also leads to telomere shortening and progressive loss of proliferation capacity. And also in budding yeast, robustness is a long-standing problem, since also in this organism, rare cells bypass senescence and re-elongate telomeres by alternative pathways. But of course, to study this phenomenon, we face the recurrent problem in telomere field, which is heterogeneity in telomere length, but also intraclonal immense differences in cell proliferation capacity. So we thus decided to identify and try to quantify the heterogeneity in replicative senescence, and we use Saccharomyces cerevisiae as a model. We rapidly realized that if one considers one single founder cell in which telomerase is deleted, it generates a progeny where all cells are different, having different telomere lengths and different cell contents. And this, so most population studies display two main biases, averaging and selection for best growers. And so we decided to circumvent these biases by analyzing single cell lineages, and by this I mean the consecutive cell divisions starting from the moment we inactivate telomerase to senescence. So we teamed up with Gilles Charvin in Strasbourg, who developed a microfluidics device which allows for the imaging of yeast cells for many days. So in this device, cells are wasted out of microcavities, so there is no limit for cell growth. And to follow individual lineages, we focused each time on the cell division right at the tip of the microcavity. In our settings, mother cells are often replaced by the daughters. So in wild-type cells, the lead we look at otherwise immortal. In this case, we use a telomerase repressible promoter, and we could see that, you just saw that we could see that upon telomerase inactivation, cells bud happily, but at some point they stop dividing, they arrested in G2M for many hours, and then they die. So it's just a repeat of the movie. Okay, but this was just one example. When we looked into tens of such lineages, we could produce this type of graph in which each lane corresponds to one movie of one single lineage, sorry. And each square corresponds to a single cell division, and the color corresponds to the duration of this cell division. And one of the striking things that appeared obvious to us was that heterogeneity was found not only in terms of lifespan, but also there was a large variety of routes to senescence. In other words, there were cells which would grow very happily up to the sudden abrupt entry into senescence, and others which experience cell cycle arrests, often consecutive normal cell cycles, before resuming normal cell cycles and back again to abnormal cell cycles. We call them type A and type B respectively. For type A, we further tested the compatibility of our results with a mathematical model developed in collaboration with the team of Marie Dunic. For this model, we took 32 telomeres out of a theoretical distribution of telomere length. We obtained, in collaboration with another mathematician, David Altman, and then we made them shorten through the DNA replication problem, which we experimentally proved a few years ago. So in budding yeast, we demonstrated that in fact the telomeres are replicated asymmetrically, resulting in two molecules, one which has the length of the parental molecule and the other shortened by the length of the overhang. And here is a simulation for the shortening of these 32 telomeres according to these molecular principles. And to cut a very long story short, we obtain the best fit when in the model, it is the first critically short telomere reaching a critical length that triggers the transition into the senescent state. And we also found that we could explain most of the heterogeneity through the initial set of telomere lengths and the asymmetry of telomere replication. To verify this in actual live cells, we developed a system to generate cells in which a single telomere is shortened to a defined length we called the final cut. The final cut is based on CRISPR-Cas9 targeted to a minimally modified telomere for this purpose. And results show that these cells, we obtain telomeres with having 80, 65, 50, or perhaps 30 base pairs. But here we've shown, I don't have time here to go into the details, but these telomeres get degraded. And this is in accordance with what we observed in microfluidics of these cells, that most of the cells have lost the capacity to grow when a single telomere of 30 base pairs was generated. And when telomeres, the short telomere is longer, we have more heterogeneity, but still we have an expected lifespan as the model predicts, except a few cells that grow a little bit more than expected. So we decided to assume that in fact the signal to senescence might be rather probabilistic than deterministic. So we decided to try a model in which, at least to explain the first arrest in type B cells, in which it would depend on the probability to arrest would increase with the shortening of telomeres. And this corresponds to an exponential growth, an exponential law of probability to describe this probability for a given telomere to signal the arrest as a function of its length. And we found that this was well describing our experimental data by better comprising the large heterogeneity of the appearance of the first arrest. We thus concluded that telomere shortening was also explaining the internal arrests present in type B cell lineages. Next, we went to look after the consecutive arrests that were found after the first arrest, and we saw that they followed a geometric law of probability, meaning that the chances of resuming normal cell cycles are constant at each cell division. And this was compatible with our recent discovery, and this is published, so I'm not going into the details. We discovered that adaptation to DNA damage in the course of senescence happens. So this adaptation corresponds to the division despite having the checkpoints on, meaning that there is telomere damage. So cells take the risk to divide despite having a problem. And combined with the possibility of DNA repair in these conditions, adaptation results in an increase of genomic instability way before the establishment of senescence. And therefore, it enables the proliferation of cells carrying mutations and genomic rearrangements. So we thus consider this mutator phenotype of telomerase inactivation a major source of heterogeneity and perhaps the ground for senescence escape. So to wrap up with the rest of the talk and to complete the whole picture of the sources of senescence heterogeneity, we add up in this picture the asymmetrical replication of telomeres and also the initial distribution of telomere length, which is very wide. And of course, we will be including in the next few years, I hope, the oxidative stress and other pathways we are currently studying in the lab. So I would like to finish here and to thank most people who have done the work. So here there are a lot of former people because we are not having any lab retreats for a while. So Zug Zhu, now he's a PI in another institute in Paris. and also Stefano Matarocci and Prisca Berardi, who have developed the Cas9, and Rachel as well, who developed the Cas9, the final cut system, and also our collaborators, mostly mathematicians. So, and our funding. So, and I will take questions.
Thanks a lot, Maria Teresa. We have already one question from Mehdi Bouhadou. I noticed upon senescence, the yeast in the fluidic device also ballooned. It increased in size. Why is this? Does this size have a limit, or do the cell lies?
Can you repeat the question? Because I was trying to get your faces back again. Sorry.
I noticed upon senescence that the yeast in the fluidic device also ballooned. It increased in size.
Yeah, balloon.
Why
Yeah,
is
they
this?
increase in size.
Does this size have a limit, or do the cells lies?
Yeah, the cells lies at some point in the end, so they increase the size. So it is typical for a DNA damage, an irreparable DNA damage, when it is applied to yeast. The cells arrest in G2M phase of the cell cycle because mitosis cannot go on. So this is the consequences of the activation of the DNA damage checkpoint. And but the cell continues to grow, so it continues to be metabolically viable, and but it's a yeast cell, so at some point it explodes, as we see. But in mammalian cells, senescence corresponds to a program. So in the end, there is a program of senescence. So the cell switches into a program, a specific genetic expression. So that this is what happens in human cells, for instance, after telomeres reaching critical lengths.
Cool, thanks a lot. There is another question from Chulong Chen. Nice talk. Did you check the heterogeneity of telomeres lengths of different chromosome ends within the same cells?
Oh, that's a very interesting question, very important one. So Liz Blackburn, a long time ago, she showed that telomere length regulation is independent among telomeres, but more recently we found that telomeres have a specific homeostasis length. So they are a little bit different lengths according to the chromosome end, but this is something. Because the sequencing of these telomeres is so hard, it has been a challenge to really have good data to determine their exact sequence and their exact length. But it's something many people are working on.
Brilliant. Thanks a lot, Maria Teresa. I think there is no more questions. Thanks a lot.
Okay, thank you.
Finish this session, I would like also to give Travis Hardcastle from Horizon Discovery the opportunity to present the company. Travis, can you try to share? Yeah, brilliant. Turn on your microphone.
There we go. Can you guys hear me?
Yes, we can hear you and see you.
Perfect. Yeah, let me start. I really want to thank Nevan and Bruno and the rest of the symposium for giving us the opportunity just to give you a brief overview of Horizon and some of the genetic tools that we can offer our customers to help expedite their research and move their research forward. And so just a brief, quick introduction of me. I am the product manager for the cell line models portfolio at Horizon Discovery. And so that includes both our off-the-shelf cell lines as well as our custom engineered cell line services portfolio. Just briefly on Horizon, if you haven't heard of us before, we are a global company who are headquartered in Cambridge, UK. That also happens to be where we perform a lot of our services, such as our cell line engineering services and our custom screening services. We also have offices that are based out of Boulder, Colorado, which is where a lot of our reagents are made. So a lot of our CRISPR-Cas9 reagents, our siRNA reagents and whatnot are made in our Boulder, Colorado site. And we have offices in Tokyo, Japan as well. What we really aim to provide are tools and services to a plethora of different types of customers, right? So global pharmaceutical companies, biotech companies, these leading academic institutes like many of you are at, as well as clinical diagnostic laboratories through our DX reagents. And we with our IP portfolio, we really take the position trying to be technology agnostic whenever we approach our customer to help approach our customers' needs and serve their needs for them. And so I would be remiss if I didn't focus on cell lines and cell line models, which is my portfolio. And so what Horizon is at core is and aims to provide is inspired cell solutions that drive the applications of gene editing and gene modulation. I think this can be seen, like I said, through the different plethora or the breadth of our portfolio, from do-it-yourself reagents for customers who want to do it themselves with the Cas9 nucleases and guide RNA, to offering all the way up to the services where we will actually do those services for our customer. Speaking specifically for our cell line engineering platform, I think we have a ton of experience. It's something that we've been doing for over 10 years now. It's something that when we first started, we were using RAAV, but we've transitioned over to using CRISPR-Cas9 predominantly these days. And if you look at some of the things that we've done, we've to date, I think we've probably had over 7,500 gene edits. We targeted well over 3,000 genes and probably used well over 100 cell lines for our clients at this point. We are really successful at doing this. We have a pretty high success rate. We feel it's floating around 94-95% success rate of being able to deliver the alterations to the cell line that our customers are wanting. And then just to highlight some of the screening services that we do, I think this is up well over 500 screenings that we've performed for our customers in-house at this point, which crosses over some of the cell lines are generated for our screening customers as well. Talking specifically about cell line products, we have a huge portfolio of off-the-shelf cell lines that we can ship in a relatively quick time, within 7 days usually. I think we're at about 7,500 knockout cell models that our customers can choose from in our half-one knockout cell line background. This is a half-load cell line that allows us to perform these edits relatively quickly and get them to our customers. And we really view these as sort of a basic entry point for customers who want to validate their hypothesis or do some quick genetic studies of validating a knockout and what that phenotype looks like. We have knockouts that are geared specifically for over 20 common research areas, Parkinson's disease, epigenetics modeling, antibody validation. We recently signed, last year, I guess at this point, signed an agreement with Human Protein Atlas where we're providing, I think in phase 1, 500 knockouts to them to validate internally a lot of their antibodies that they're producing, and there's an opportunity for a phase 2, which will provide 500 more cell lines to them for the half ones for them to validate their antibodies. And I think I've overheard and listened to some of the talks that the previous symposium was on cancer, specifically focused on cancer. We do have several cancer-related cell lines off the shelf that are available too. I think we're at 300 knockouts or knock-ins, and many of the common, well-studied mutations or many of the common cell line backgrounds, and those can be of extreme value to our customers. Recently, we launched our Cas9 stable cell lines. This is both our Cas9 randomly integrated into many of the common cell lines, both Cas9 as well as our dCas9 BPR reagent for activation types of studies. There's a wide range of cell lines we offer with that, and it really offers customers the opportunity to do both gain and loss of function studies in the same cell line background relatively quickly. I'm running a little short on time, but I just wanted to highlight, I'd be remiss if I didn't touch base on our services. We really take pride in being with our customers from beginning to end, from consultation to delivery at the end. That goes from the very beginning when our project management team reaches out for the consultation and provides a detailed work plan. We are, constantly improving upon this. We have a tool that we're launching here relatively quickly online, where a lot of that technical feasibility will be addressed by AI with our online tool that will help customers design projects relatively quickly. And then we provide updates along through this whole process and the pipeline, from reagent designs to cell line onboarding to gene targeting, through the single cell cloning and on into the delivery phase of the project through the duration. Just a quick case study of what we're seeing a lot of stuff in. As you know, biology is complicated. We're talking a lot about that these days in the symposium, and we're seeing a lot of these more complicated types of projects come through, with a lot of customers wanting to edit in iPSCs as well as primary cells. And so that's something we're really well at doing. We have, I think, completed over 75 projects today in these complicated cells or primary cells. There's some of the data of, some of the validation we provide from, many of the pluripotency stains, AP staining, doing CNV analysis and karyotyping. And we've done a wide plethora of types of projects in IPSCs. And one last thing before I get out of the way and we can get on with the rest of the great sciences being presented is I focused on cell lines, but we do have a ton of other great genetic tools that can be used by scientists. Our edited gene editing reagents. We have pre-designed guide RNAs. Plethora of touch base a little bit on solutions that are powered by Darwicon, as well as our CRISPR reagents, which will be coming out in the next month or two, and as well as our CDA and ORF collections, and as well as screening services on top of our cell line engineering services. If anybody has any questions, please feel free to reach out to me, and I'll be more than happy to answer those. And with that, I will get out of the way, and we can get on with more of the fun science that we would learn about.
Thanks a lot, Travis.
Yeah.
That please maybe conclude this fun. Thanks to everyone for participating. It's a break. We'll start again at 9:15 AM San Francisco time, 6:15 PM Paris time.
Maybe we start again. So Clotilde, can we see you and can you share your screen? So we will start with Clotilde Policard. She's professor at the Ecole Normale Supérieure.
Can you see me?
Yes, we see you.
Okay.
Can you share your screen?
You have to unmute me. I can't speak.
It's not me who
okay.
has the control over that.
We can hear you just fine.
Okay, you can hear me. Okay. Okay, so I activate share my screen. Is that it? Okay.
And so people should have a question in the Q&A while Clotilde is sharing her screen. Good.
Sorry. Do you see my screen here? Okay, and here.
Perfect.
Okay.
We all hear.
Just 1 minute. I take the pointer. Okay. Okay. Well, thank you very much for. Oops. Can you see everything, or is there something blocking here? No. Is that okay for you?
No, it's fine.
Okay. Okay. So I'm really glad to be here, if I can say that today, to present you what the work that we are doing at the Ecole Normale Supérieure. So we are chemists, but we are so inorganic chemists. We work with metal ions, but we are very much interested, as I'm going to show you, with the way that they, with their fate in a biological environment. And what I'm going to present you today are we are interested in mimicking metalloproteins that can be involved in important processes such as antioxidant protection. And you will see that what we are doing is connected to inflammatory bowel disease. So we have already seen a presentation about that and with the fate of this molecule in the gut possibly. So these are the people that participated to the work. So Nicolas Delsuc and Hélène Bertrand, that are my very close collaborators. Here, the students that did the work I'm going to show you, and our collaborators. So we work in close collaboration with a group of medical doctors from the Hôpital Saint-Antoine and with Joël Vin at ESPCI, which is very close to ENS and Institut Curie. And so we have just obtained a funding for a project that I'm going to show you for which I'm going to show you some preliminary data with Joël, and that was funded by QLife. So we work on superoxide dismutases. So these proteins are very interesting because they are very efficient. And because of that, they have been studied to understand their efficiency. They catalyze the dismutation of superoxide, which is the first reductive, let's say, molecule from the oxygen. So when the oxygen is reduced by 1 electron, it gives superoxide. And so these proteins are meant to catalyze the dismutation of superoxide at a very fast kinetics that is close to the diffusion limit. So spontaneously it would disappear, but when the oxygen is reduced by 1 electron, it gives superoxide. And so these proteins are meant to catalyze the dismutation of superoxide at a very fast kinetics that is close to the diffusion limit. So spontaneously it would disappear, but but with a kinetic that is too small to maintain low concentration in cells. And so what is oxidative stress is not the production of superoxide or reactive oxygen species, because they are continuously produced by natural metabolism, let's say. It's more a rupture of the balance between ROS production and antioxidant defenses. And when this occurs, so the reactive oxygen species may react with many biomolecules, such as DNA, lipids, or proteins. And so superoxide dismutase are a very large family. They are all metalloproteins, or using or copper, or manganese, or iron, or nickel, as shown here at the active site of superoxide dismutase. What is interesting is that these are different metal ions with different redox properties, but they do the same function. And what we do, we try to design low molecular West complexing to reproduce the activity of this protein and to be used in cell systems. So we focus at the active site of this protein, and we try to reproduce the main parameter that is responsible for this very good activity, which is the redox potential that is organized, or that is, let's say, set up by the overall protein that is organized around the metal ion, so that the redox potential of all these metal ions at the active site of SOD are set up at the same value. Whereas if they are not in this environment, in this proteic environment, they will show very, very different redox properties. So in the lab, we develop peptides using combinatorial strategy to perform this activity. We also develop, in collaboration with a group at University of Michigan, let's say, kind of maquettes with a designed active site for metal ion. And we also design low molecular weight complexes. And today, this is what I'm going to show you about, and how we study the activity of these molecules inside a cellular environment. So we are interested in inflammatory bowel disease because they are associated with oxidative stress, and with what is very interesting for us, with a weakening of the anti-superoxide defenses. What has been shown is that in Crohn's disease, the MNSOD, which is in our mitochondria, is overexpressed but in an inactive form, whereas copper SOD, which is cytosolic, is underexpressed. So that means that there is not enough SOD to control the flow of reactive oxygen species, and particularly superoxide, which is at the origin also of other reactive oxygen species, such as H2O2. And so in collaboration with Philippe Sexic and Sylvie Dominiot at Hôpital Saint-Antoine, we use epithelial cells that have been stably transfected to be very sensitive to lipopolysaccharide by producing MD2, which is a co-receptor for LPS, which is a soluble protein, the co-receptor of LPS, and that will make these cells very, very sensitive to bacterial LPS with high level of inflammatory markers that we can follow. And our question is, if we incubate these cells with LPS and this molecule, can we reduce the inflammation induced by LPS, which is supposed to be mediated by oxidative stress? Okay, so we think that this is very important to characterize the intrinsic activity of this molecule, but also the activity in cells. So what we do when we have such a molecule that is active in assay tube, we check, we try to quantify the quantity in cells. So here, this is using EPR, the signature of manganese here, but we can also use ICP-MS. We also try to follow the fate of this molecule. Can we track it using MSMS, and what is the speciation in cells? We also try to characterize the distribution. Is the molecule distributed everywhere, which is the case here, or is there a specific location? And for that, we use a direct signature from the manganese using X fluorescence, which is a non-conventional imaging, but yet you can perform on cells. And these cells here have been cryofixed. And then we also characterize the bioactivity. So here I just show what we obtain with EL8. So these are control cells. When we activate them with LPS, we have a very strong increase in the EL8 because they have this MD2 mutation. And when we co-incubate LPS with MN1, we see that there's a very, very strong reduction of this inflammation. And then we wonder, is there a link of this, is there reduction associated with a change in the native anti-superoxide protection? So we look at the copper SOD, and as you can see, with all these conditions, and I have also here including MnCl2, I should have left it here as well, I forgot to do that, but MnCl2 is not active to reduce inflammation. And what we see here is that when we have, for copper SOD, there is no modification of the copper SOD expression. But for MnSOD, there's a very interesting behavior because LPS increases the amount of MnSOD after 6 hour incubation of LPS. This is because this is associated with the presence of oxidative stress. And here, when you have co-incubation with MN1, you see that the cell does not need to overexpress the MnSOD, whereas MnCl2 is not active. And so then we wondered, is it possible? So we've seen that with copper SOD and MnSOD. Can we have a proteome-wide characterization of this, of the general behavior of LPS activation and MN1 activation? So what we perform is a SILAC method to perform a quantitative titration of the proteome and see what is the effect of LPS and what is the effect of LPS plus MN1. And we've performed that with fractionation to increase the sensitivity at the mitochondria, which we think is the main organelle. And from this kind of experiment, I can discuss that if you wish, we can extract something that is called a volcanoplot and something that is telling us something about the redox status. So very quickly, what we see with the preliminary data that we have here, we see that LPS is associated with an increase in oxidative stress, which is resolved at 6 hours. And MN1 is limiting strongly this redox effect, which is interesting, showing that MN1 shows its antioxidant activity in the cells. And now if we look at the volcanoplot, we see what is interesting is that at 15 minutes, so very, very short, there are some modification at the mitochondria, which could be associated with trafficking of proteins, maybe not with protein synthesis. But this is not the case with MN1. And at 6 hours, we have a range of protein that have been modified that we can discuss if you're interested. But what is interesting is that MN1 reversed the effect of LPS on the proteome, so which I think is quite interesting. And so we have shown that this effect can also be seen on mice. And sorry if I'm going a little bit out of time. So this is the summary of what I'm trying to tell you about. The people that I want to thank and you for your kind attention. Thank you very much.
Thanks a lot, Clotilde. That was really interesting. So do we have question? I don't see question yet. So maybe before we have more question, I can ask one. So just by curiosity, what is exactly this X fluorescence that you were mentioning?
Oh, okay. So this X fluorescence is just, so we do that on synchrotrons and we shed a very energetic light, which is about 8 kilo electron volts, and we scan the cell. So maybe I can go back to this. Okay, and we scan the cell here. So each pixel is about 200 nanometers, and what we see is the distribution of all heavy elements. So potassium here is an example showing, because potassium has an even distribution over the cell. So what you see here is something that is thicker at the center, which is probably the nucleus, and something flatter on the periphery. And here we see we have the information about manganese. And so we have information also about other metal ions like iron, copper, or well, any metal ion that, calcium, of course, any metal ion that we want to look at. So it's a non-conventional imaging because it's not readily accessible. You need to go on a synchrotron to have a very focused beam, but then you can have the information on the distribution of heavy elements. And we perform that on cryofixed cells because we don't want the soluble species to go away when you fix them chemically.
Okay, thanks. Are there more questions? I don't see question yet. So maybe, yeah. So you are able to follow actually also your molecule inside the cell from this. This is what I see from your scheme.
You mean from the image here?
No. the thing above. So
which one? Ah, this one?
Here? Yeah, yeah.
So we do that. We do that on cell lysates, and we try to track the signature of the molecule by MSMS. So this one is the MSMS signature. So the mass of the molecule is 408, but then it fragments one more time with the specific signature, and so we can tell that the molecule is there. So now we're trying to convert that into something where we can do also quantification to see how, so it's there, but how much of the molecule is there, we don't know. We know how much we have put for the incubation, but knowing how much has been entered is something different. And so this is something that we are trying to do now.
Okay, so now
real quantification of this species.
Okay, yeah. Two questions. So from Nevan, can you comment on the post-translational modification involved in your proteomic?
Oh, you mean, so what we look at is we look at mainly, I don't know if that is your question, but we look at so we do, what I didn't say because I was too fast, is that we perform both proteomic and redoxomic, and we look at the tiols, and we look specifically at the SS bridge versus the SH. We look at the cysteine that have been oxidized and the cysteine that are reduced, and this is how we try to track this redox status of the cell. So these are the PTMs we're looking at specifically in this method, which is redoxomic, and this has been set up by Joëlle Vin, and so she's doing that in many different
He was rather mentioning phosphorylation or ubiquitination.
We have not looked at that yet. Yeah.
Okay, and maybe a last question from Olivier Espeli. Olivier, can you spell your question because I don't understand it even when I read it? Can you?
Sorry, I can't see the. Maybe I can see the screen now.
Couple this X-fluo with TEM or epifluo. This X-fluorescence with TEM or epifluorescence.
Yeah. Okay. So we do that. When we do this imaging here. We know now how to, where is it? When we do this kind of imaging, we're able to track these cells, for instance, and look at it in classical fluorescence. So this is possible, and this is something we do because not with this molecule, but with other molecules, we have a multimodality, so we can look simultaneously at the molecule in X-fluorescence and in classical fluorescence. We've tried them, we've never obtained publishable results, but this is something possible. It's, yeah.
Okay. Thank you. So thanks a lot, Clotilde. Beautiful. Okay. You're on.
All right. Thank you very much. I want to thank Nevan and the organizers for the opportunity to tell you about some, very new and what we think is very exciting work that we've been doing in the lab. And it should be, well appreciated that protein homeostasis is a process that's vital for all organisms. Central to this process are molecules known as molecular chaperones. Many of these are ATP-driven complex machines that help, facilitate protein folding, but also degradation and avoiding aggregation. Now, our work is focused on one such molecular chaperone. And one of the reasons is that this chaperone known as HSP90 is persistently required throughout many of its protein substrates lifetime and not just during initial folding. And not only that, but HSP90, instead of working with all proteins, is focused on a subset of proteins that are highly enriched in signaling proteins such as kinases, transcription factors, et cetera. And as a consequence, HSP90 has a profound influence on cell biology and developmental biology. This persistent interaction, we think, implies a role not only in client stabilization, but also in regulation. And so our effort and how this general machine works, how it chooses what clients to work on, this 10% of the protein, and in atomic detail, how does it function? And so for today, I'm going to focus on the work of our studies on one particular class of clients typified by the glucocorticoid receptor. This is an obligate HSP90 client, and it's a ligand-activated transcription factor that lives in the cytosol, and then when its ligand cortisol comes in, the active receptor then translocates into nucleus, where it drives a very complex transcriptional program. Pioneering work by Sue Lindquist and Keith Yamamoto and their labs in the 1990s, really, that this process of activation in the cell doesn't happen alone, but in fact requires a complex array of molecular chaperones, including HSP70 and its co-chaperone, HSP40, and HSP90 and two of its co-chaperones, HOP and NP23. And so the key part that is involved in interacting with the chaperones is this ligand binding domain that binds the cortisol. And you can see from this crystal structure that the cortisol ligand is buried deep within the protein. In fact, if this were all space filling, you wouldn't be able to see the ligand at all because it's completely buried. And so the concept has generally been that, because this is so large and hydrophobic, that a lot of the stabilization of the molecule comes from hydrophobic interactions with the ligand itself, and hence it needs chaperone interactions to stabilize it. But building on the fact that we knew that these components were required, we wanted to then use this as a basis for studying this in vitro through a complete reconstitution and really try to get at the mechanistic underpinnings of this process. So the initial work started with inactive GR, but Elaine Kirschke, a graduate student in the lab, turned this around and started with active GR. And from this, she was able to dissect the very surprising finding that the HSP70 component, which is indeed part of this necessary requirement to get to the active state, instead of just playing an activation role, will take active GR, that is GR that either has ligand bound or can bind ligand, and inactivate it and completely turn off the ability to bind ligand, as shown in this equilibrium ligand binding study. Now, if you add in HSP90 and its components, you can then take this inactivated material and restore full ligand binding capability. And so one of the surprising findings of this study was that much of the HSP90 dependence of the glucocorticoid receptor is really tied to this inactivation that happens by HSP70. And so if we consider what's going on in the cell, there's really a constant cycle going on driven by the ATPases on HSP70 and HSP90, where a glucocorticoid receptor is inactivated via complexing with HSP70. That then transits to form a loading complex involving 70 and 90 and a co-chaperone. On ATP hydrolysis on HSP90, this then transforms into a maturation complex, at which point the glucocorticoid receptor is now active for function. And then ultimately free GR can be released. And the remarkable aspect of this is that the affinity, that is the ability to bind ligand, the kinetics, are all controlled as much by the ATPases of the chaperone cycle as they are by the intrinsic properties of the glucocorticoid receptor. And so in steady state within the cell, you never have a constitutively active receptor, but instead it's being turned on and off constantly, driven by where any molecule is within this cycle. And so our goal really has been to understand the how of this process. How does 70 inactivate it? What's the nature of this loading complex, to give us clues as to how it then ultimately becomes reactivated in the maturation process. And so very recently, we've made great progress on this through cryo-EM studies of these various structures. Very talented postdoc, and there are many, many surprising features. Virtually everything we see was completely unexpected, but just to highlight a couple of points because of the lack of time, that instead of one HSP70 involved in this complex, instead there are two. This one over here comes in and delivers the glucocorticoid receptor via having the receptor being bound to its substrate binding domain. This HSP70 is primarily playing a scaffolding role to buttress this co-chaperone hop. Hop in turn interacts with the receptor to provide a binding site that I'll tell you about. But the combination of the 70s and the hop interactions together poise HSP90 in a conformation that's ready for ATP hydrolysis, but yet blocked from it at this stage. If we zoom in and take a perspective focus on the GR, we can see that the glucocorticoid receptor is threaded through this complex lumen of HSP90, hop, and then binds to HSP70. And together, this defines a very extensive and complex binding site that contains hydrophobic elements from HSP70, other kinds of hydrophobic elements from the co-chaperone hop, and then polar and hydrophobic components from HSP90. And we think that this extensive binding site and these patterns that are set up really distinguishes client from non-client interactions. And so how does HSP90 activate GR in the maturation state? This is the work of a superb graduate student, Sherry Noddings, who determined a very high resolution structure of this complex. And it's stabilized by P23 that not only stabilizes the 90 conformation, but makes completely unexpected but direct interactions with the glucocorticoid receptor. If we zoom in to the focusing on the glucocorticoid receptor, it too has a region going through the lumen of this closed HSP90, but now this is the region that was previously bound to HSP70. That's critical because by sliding that region into the lumen of HSP90, this helix 1 that was interacting with the co-chaperone now has a chance to refold against the body of the glucocorticoid receptor, and in fact restore a completely native receptor that binds ligand. And so from these top-down views, we can see that very clearly, where this region that was bound in HSP70 now slides through, allowing this helix to refold there. And so sliding is a key aspect of this mechanism, and it's driven by conformational changes in 90 that are taking place during this transition. Sliding goes beyond just those motions, and in fact allows HSP90 to work processively on attached domains. And so, for example, in our particular project, we had MBP attached as a fusion protein to the N-terminus of the LBD. And in addition to the structure of GR that I just showed you in the maturation complex, there's also a set of molecules that correspond to MBP, but this is now on the opposite side of where GR was. And what's happened is that engagement started with the glucocorticoid receptor, and then this slid through such that now the C-terminal part of MBP, the last two helices, become unfolded and inactivated. So this allows the to work. Emphasize in the end here is that different clients can utilize the HSP90 cycle in very, very functionally different ways. And so, for example, whereas I showed you with glucocorticoid receptor, this state, this maturation state gave rise to a completely active and functional glucocorticoid receptor. In the case of a kinase, a structure that we saw a number of years ago, that the kinase is completely inactive because it's been ripped apart and the active site destroyed. And we can see that very clearly here. And so what this means is that the activity that results in this final state of the 90 cycle is dependent upon where this binding sequence is placed compared to structural elements within the client protein. And so if it's at a place like this, then you can get a native protein. If it's here, it's completely inactive. And so that the functional outcome then becomes an evolutionary choice for the client. Finally, I want to pick up on something that Sophie Dumont brought up yesterday, where we're talking about just general concepts of regulation and where this requires tight cycles of opposing reactions. And so phosphorylation and dephosphorylation, Sumulation and desumulation, even transcriptional regulation only works because it's opposed by degradation. And what we're proposing is that there's another whole class of completely reversible regulatory processes that are driven by or based on chaperone-mediated activation and inactivation processes that are completely reversible and provide orthogonal regulation strategies. And finally, I want to leave you with the thought that this coupling to the chaperone machinery has become an evolutionary choice as opposed to a biophysical necessity. And so I want to end, of course, with thanking the superb group of people that's been involved in all this work, highlighted our Ray postdoc, graduate student Shari, that really did all of this GR work that I told you about. And I'll stop there.
Thank you, David. Fantastic talk. So you have many questions already. So maybe I start with a question from the audience, from Mehdi Bouadou. So of course, he's asking the question of phosphorylation, for example, so post-translational modification that would control the interaction between the glucocorticoid, the GR and HASP70 or HASP90. Have you?
Yes, so there's a huge number of PTMs of all kinds, phosphorylation, acetylation, methylations, on all the chaperone components. And the thought is, and from work from other labs, is that these likely tune the responses in very complex ways. And especially like they show up in the case of cancer cells that can have very different properties of their chaperone system that has been remodeled to deal with the cellular necessities. We have not begun to really dissect that yet in detail. But one of the interesting thoughts is that the conformation of the client that's bound to HSP90 may actually provide a framework on which modifications can be put on and taken off on the client that are different than the native state of the client.
Okay, thanks.
But great question.
And so question from the audience, from Nina. So do you thoughts about artificial intelligence?
There's recently, out of the Google team, just recently, there's been some, fabulous report on progress that they've made in prediction of protein structure just from sequence and other information like, other structures and things. And it was stunning. They completely blew away the recent, CASP competition, which is designed to do this. And I think it'll have phenomenal implications for structure biology going forward, and so, it really speaks to being able to do a decent job of determining structures of individual molecules. I think experimental approaches will still be very good for looking at, conformational heterogeneity, different conformational states, and especially complexes. But it does pave the way to be able to then do much more in the way of identifying structures within cells via cryo-EM tomography. And docking in structures of individual molecules that have all been determined computationally. So, the field hasn't begun to grapple with the implications of this, but it is very, very exciting. Great question, Nina.
Okay, thanks. I think for the sake of time, you have two other questions from the speakers, so maybe you can answer them directly, and we will move on to the next speaker. Thanks a lot, David.
Okay, thank you very much.
We will move on to William De Grado. He is also professor at UCSF in QBI also. So can you share your screen, William?
Okay. So is this working?
Yeah, it is. Thanks.
Okay. All right. So what I'd like to talk about today is de novo protein design. It's really quite different from what we've been looking at until now, in that a good biologist or biochemist will try to start with a complex biological phenomenon and reconcile it to the principles we've learned from physical chemistry, and what we're doing here is starting with first principles and trying to build up proteins that are really quite complex. Why do we want to design proteins from scratch rather than modifying natural proteins? Well, first, it's our fundamental understanding. If we understand proteins, we really should be able to design them entirely from scratch. Having done that, we can then begin to design proteins with desirable functions. So I'd like to talk to you about some advances we've made today in the hopes that perhaps it would lead to some collaborations or interactions. So what we're going to focus on is a particularly difficult part of de novo protein design field, which is the design of proteins that bind specifically to small molecules and cofactors. So this is really the inverse of drug design, in which one starts with a natural protein, tries to make a small molecule that binds to it. Instead, what we're doing here is starting with a small molecule that is our target and trying to design a protein that encompasses it. And so the question is, can we devise methods that really work without requiring building large libraries and screening on yeast or phage? Now, the beginning point for all of de novo protein design is really the Anfinsen hypothesis, which states basically that the sequence in the right milieu codes for the right three-dimensional structure, dynamics, and ultimately function. What we do in de novo design is just the reverse. We start with a three-dimensional structure, which we want to design, and then we search for a sequence using side chain repacking algorithms that will stabilize that structure in water. But this is only half of the Anfinsen hypothesis. The other half is even more challenging, which is function. And in some cases, we can start with a de novo designed scaffold and introduce function. That three-helix bundle I just showed you was used extensively by Vince Pecoraro's lab, who we heard about in a previous slide for metalloproteins. But what we'd really like to do as we get to more complex functions, and this has really become necessary for small molecule binding, is really start with the function, let that dictate the tertiary structure, and then as before, we're going to go and find the sequence. Let me give you an example of how we do this, starting with bioinorganic chemistry. And this is one of our targets. We wanted to design a cuboidal system not seen in nature, consisting of zinc atoms. And the ligation to the metal ions now is going to dictate, if we go backwards, the backbone conformation, in this case, a 4-helix bundle. Additionally, we start building in second shell and third shell hydrogen bonds to really fine-tune the geometry. But what's really critical is what's going to hold all this together and enable the folding. And this is what we call the core principle, in that we have hydrophobic cores that very precisely position the business end of a protein so that it can bind and have the appropriate dynamics. And we found in de novo design, to achieve this, we really needed to design not only the binding site divorced from the core, but we had to design the entire protein entirely in one computation. When we did that, then we started to succeed in a variety of applications. Before leaving this inorganic chemistry, I should mention also that we can design proteins that can self-assemble in the presence of metal ions. And these could be used for triggered assembly for either chemically induced dimerization or phased separation. So now let's talk about designing the organic molecules, and the magnitude of the problem is shown here by a recent paper from David Baker's lab, in which he designed a protein that binds to this flat hydrophobic dye. Despite the simplicity of the ligand, It really took a large number of very talented coworkers. It was a landmark paper. Dozens of proteins needed to be prepared, structurally looked at. Ultimately, they found some mid-micromolar binders. And then after extensive optimization by yeast display, they reached something that was sub-micromolar and could be structurally characterized as a complex. The question is this all necessary? Could we go directly from a computational design with high fidelity to experimentally verified binders without extensive screening? And also, could we build proteins that bind much more complex and useful molecules? And can we achieve this success without assembling very large teams of scientists? So how does, the first thing we did was to step a little bit back and say, how does nature evolve small molecule binders? And what nature does is it explores sequence space sampled in the context of a backbone fold and finds local sequence and structural motifs that provide convergent interactions to bind polar fragments. For example, here from Tawfig's lab, we're seeing the places where phosphate can bind, and there are a number of different motifs, but in each of the certain sequences and three-dimensional structures that pair now and allow us to position the phosphate with good energetics and geometries. So our hypothesis is that we can find privileged positions to bind polar groups through multivalent and convergent interactions. In order to do this, though, in a general sense, we really needed to develop a new metric of protein structure, which is related to rotamers. Rotamers define the conformations of a given amino acid, in this case, aspartic acid. And if we know the backbone conformation here, then it allows us in de novo design to say exactly where the side chain is going to be when it's in one of its rotameric conformations. Here we instead ask, given a backbone, where is an interacting group located? And can we find favorable interacting groups through database searches? And in fact, we did. So these are the places the carboxamide could bind. It's hardly a continuum, it's quite punctate. And we also see the distributions depend on secondary structure, here for a helix and here for a sheet. And let me show you how we use these then to make congruent sites. Let's say we've got an alpha helix here. Now we can use Vandermeers from a number of different proteins, and we can assemble them on the helix. Let's start with an asparagine, bring a carboxamide, and then down over here, a histidine takes the carboxamide into precisely the same spot. And a serine and a threonine, and all together, they can converge. Doing this computationally has been quite a challenge, but this is a convergent site. Now, if that's all we want to do, we'd be done, but we want to design much more complex structures as shown here. And so how do we fulfill all the binding sites within a drug like this? So our first drug that we approached is apixaban. It was suggested to us by a cardiologist because of a need to turn this blood thinner off prior to surgery. It's a real difficult challenge in that we have 5 rotatable bonds and 9 polar heteroatoms to deal with. We divide it then into fragments that we want to bind, and then we place those fragments relative to helical bundle backbones using Vandermeers. And what that allows us then to do is to find binding sites within a given backbone that are consistent with fulfilling the hydrogen bonding potential of that drug. Now, we use parametric design, which we developed a number of years ago, to cycle not through one helical backbone, but a variety of helical bundles. So we can change the radius and the phase and the major and minor phase and the helix offset. In fact, we do these simultaneously in search for a conformation that allows us to bind. And so doing this, we designed 6 proteins that were tested. 2 of these worked from the criterion of binding tightly and specifically, and did not require phage or yeast display. This is the crystallographic structure of what we found. In fact, everything's positioned precisely as we had anticipated. We also did mutagenesis and looked at related compounds to show that it was binding specifically. Finally, at a 1.3 angstrom resolution, we can see in the apostructure a water network that is completely displaced when the drug binds here. So what do we want to do with this? Well, this could be a nice targeted delivery vehicle for delivering small molecule drugs where we want them. Could be used as reversal agents and antidotes for small molecule drugs. And we could easily imagine making chemically induced dimerization and oligomerization systems. So thank you very much for your time. And my coworkers are shown here. Almost all of this was the work of Nick Polizzi, one person who developed the approach and did all the experiments. Xiaoxing Zhang did the work on di-zinc or tetrazinc structures in collaboration with our University of Naples group.
Thanks a lot, William. So we have questions from the audience. So how do you think AlphaFold may change your approach to designing de novo proteins? This is from Maya Modak.
So we are already using approaches like that, but not as sophisticated or as good. But clearly what it allows us to do is to test at the apo state whether our proteins bind into the desired structure. So do they fold right? And it'll be more effort to actually see whether the binding sites occur using deep learning approaches like that. But it's already impacting greatly. Yeah.
So another question from Monita Muralidaran. How do you experimentally analyze the dynamics of the small molecule and synthetic protein binding, and how do you extrapolate it to physiological conditions?
How do we evaluate dynamics of binding? Clearly, we need to have a binding site available. When we're trying to make something that's catalytically active, then it has to be very easy to get in and out. If we want to make something that's a carrier, then we may want different degrees of burial. So that's slower on rate, slower off rate to achieve the biological purpose. In terms of biological milieu, what do we want to do? If we want it to be degraded, then we want to design the protein out of L amino acids. If we want it to be stable and non-immunogenic, so it's not presented by MHC in fragments, then we can make it out of all D amino acids.
So from Alicia Richards, once assembled, are there any unique challenges for de novo protein to elicit the wanted responses?
I'm sorry, what?
Once assembled, so in a way, are these de novo protein behaving differently than natural protein? I guess that's the idea behind this question.
I don't imagine so. Hannah showed us some beautiful examples yesterday in collaboration with the Baker Lab, in which they're looking at feedback circuits that are doing a beautiful job of mimicking nature. One more thing I want to make a plug for is not giving up sort of the atomic level, molecular level approaches to design in favor of parameterized approaches that are based on informatics. Because if we ultimately want to design non-protein molecules for useful purposes, we really need to have an atomic level understanding.
Okay, so you have many other questions, both in the Q&A and for the speakers. So if you can answer them directly, thanks a lot for your impressive talk. And
cool.
we'll move on to the last speaker of this meeting. Thanks.
Thank you.
of the bacteria. And there we treat very well defined and localized types of cancer. It is already approved to treat really different types of cancer, as you can see here, but also now infection like sinusitis. However, the drugs, the photosensitizer, which are currently on the market, I think there are about 13, have quite often the same troubles. It means they are poorly water soluble. The synthesis to make those compounds, it is very difficult to do. For the patient, normally after treatment, they have to stay with long clothes, long sleeve clothes, because they are extremely photosensitive. The photosensitizer have a tendency to accumulate in the skin, so they will get actually sunburns, which is, for example, a problem in California. Also, the compound is very has a low selectivity, I would say, for cancer cells. That's why you would like to prepare a new generation of compound which will go better to cancer cells and which will be easier to make and much more water soluble. And this is why when we started our independent career, that we have decided to use metal complexes like other people in the field. And why we have decided to use such type of a complex that we call ruthenium-2 polypyridyl complexes is because they have all what you need to have in order to have a perfect photosensitizer. They produce singlet oxygen, the killing agent in PDT. They are not photobleaching. It means when you produce the singlet oxygen will not destroy the photosensitizer. They are very easy to make. They are relatively highly water soluble because you have a positive charge here. They are inert, relatively non-toxic. It has already been demonstrated this, and relatively cheap to make because the synthesis to make those compounds is extremely short, a few steps. Now the highlight, and I'm very happy. Last week, the FDA has given a fast-track approval for this compound, which is called TLD1433. It's for my colleague in Texas, Sherry McFarland. And this compound is a PDT photosensitizer to treat bladder cancer. Bladder cancer is a very nasty type of cancer because quite often it involves the removal of the bladder, which is really terrible for the patient. With this compound, you can directly put it into the bladder through the urethra. You fill first the bladder with water. The compound has a tendency to accumulate into the cancer cell of the bladder, and then through fiber optics through the urethra, you can actually save the bladder and really save the patient. And it is very nice to see that such a simple to make compound will maybe soon be approved by the FDA. And this is really an encouragement to keep working in this field of research. Why is that? It is because for the moment, the compound like this one a low bladder cancer, like TLD1433, green light is used. It means you are using 500 roughly nanometer, which is normal because bladder is extremely thin and you don't want to go through the bladder with the light. However, if you want to treat deep-seated tumor or large tumor, you need to use wavelengths with a higher wavelength at about 600 to 800 nanometer. But in order to do this, you need to use other types of ruthenium polypyridyl complex. And this is what we wanted to do in this project. What we have decided to use as a technique is called two-photon photodynamic therapy. Instead of using, for example, one photon at 400 nanometer to reach the excited singlet state, which will undergo afterwards intersystem crossing to reach a triplet state, and this triplet state will be able to react with the triplet oxygen in your tissue to produce singlet oxygen. You can actually use, if you use a laser which is very powerful, you can use two photons at 800 nanometer to reach the excited singlet state. And this is very interesting because with 800 nanometer, you can go really deep into tissues. What we also need is very dedicated students, and I would like to thank Johannes, who is now a postdoc in San Diego, France, who went back to Switzerland, and Marta, who was co-supervised with Bruno, who went back now to Poland, and they have done all the work that I will present today. What we have done in this project is to work with ruthenium complexes, which will have the ability to absorb 2 photons. And what we have done is to team up with Ilaria Ciofini from ChimieParisTech to do TD-DFT calculation to predict which compound will be the best. So before doing a lot of synthesis, we have done first computational studies. And you will see that we have decided to go for different types of ruthenium, which are either symmetrical or non-symmetrical. Right, it's going with. So what you can see is compound 7 was extremely potent, okay? And this is why we have decided to use it for all the other experiment. So this is compound 7. It seems that I have not been given a lot of time today. So what you can see with confocal microscopy is that it is going into the cytoplasm. And what is very nice in the context of QLife is because we have ruthenium complexes, we can actually use ICP-MS also to quantify where the compound is going. So by looking at the ruthenium, which is not present in normal cell, you can see that the majority of the ruthenium is indeed going into the cytoplasm. You extract organelles after organelles. What I want to show you is that our compound is indeed producing singlet oxygen. We will use this dye, we have the cells, we have put a compound, shine light, and you can see that indeed the fluorescence is happening in the cytoplasm. You can also see, you can also see that the compound is producing a mixture of paraptosis and apoptosis when you shine light. This is what is happening. We have the compound in the dark, it is fine, we shine light, and the cells are really blasting. What we have also used is this spheroid, this assembly of a cancer cell to form mini tumor. And what is interesting is because we have 2 photons, we can go really through the tumor. And you can see now that the compound is going even in the core of the tumor. What you can also see after 3 days, if we let the spheroid grow, we shine one slide, you can directly see that compound 7 can really destroy with 1 photon or 2 photons the mini tumor. And very importantly, to finish up, we have done also in vivo. You can see that we can inject the compound, give light. There is no loss in weight for the mice, meaning that the compound are non-toxic. And if we shine light with 1 photon or 2 photons, we have really a serious reduction of the tumor size. As you can see here, this is with 1 photon, with 2 photons, compared to the control. The technique is working extremely well. I just hope that over these minutes I could convince you that ruthenium complexes are excellent PDT photosensitizer. They can use for both 1 and 2 photons. And we have even now shown that we can encapsulate the compound into polymer approved by the FDA to have even a higher tumor accumulation. Thanks a lot for all the people in Paris working on this project, all the people who were working in Zurich, and the people who are collaborating with us in this project, and finally the people who have given us money. Thanks a lot for giving me the opportunity to talk today.
Thanks a lot, Gilles. Very interesting talk. So we don't have yet question, but maybe I have one. So how, maybe I missed it, but so how does a compound function to actually limit the growth of tumors? I completely missed the point, I'm sorry.
No, that's not the problem, you know, I went very fast. Actually, the compound you shine light, it oxygen that you have in tissue, and this singlet oxygen is extremely toxic because it will oxidize everything into the tumor in the cells, and this will lead to a paraptosis or apoptosis. And this is very interesting because singlet oxygen is extremely short-lived, so you will have really a reaction only on the site of irradiation. So it allows like a chemotherapy which is extremely localized, because the compound in the dark is non-toxic. And this is really the great advantage of PDT.
Okay, so there is a question now. So from Maya Modak, does this work with different kinds of tumor?
Yes, we have really used different types of tumor, and this is really the nice part of this technique, because even normally we never observe any resistance, because as soon as you produce singlet oxygen, it is a photocatalytic process. As long as you produce, as long as you irradiate, the tumor, and there is a photosensitizer in the tumor, you will kill and kill and kill. And this is one of the great advantages of this technique. So you can really thinking about killing any types of tumor as long as you bring the compound to the tumor. Once again, I'm also very honest, it is really for localized types of cancer.
Okay, so there is a question from Monita Murali-Daran. So does the reactive oxygen species affect the normal cells?
Well, yes, for sure. But the point here is you only irradiate at the site where you have the tumor. And this is also why now that we are really trying to improve the accumulation of the photosensitizer in the tumor. You have a few techniques to do this. For example, by encapsulation, we have seen that you can encapsulate into a polymer with biotin, for example, and biotin is highly required by tumor. And this is why actually you will really mainly kill only cancer cells. I hope it is clear how I have replied to this question.
So there is a question from William Degrado. So what strategies used to increase low IS?
So this is a very, very interesting question, and this is why we are working with heavy metals in order to increase the spin off, and we are currently even now trying to use heavier metals in order to not only increase the lifetime, but also to go longer wavelength upon. And what you have also not to forget is that the compounds are phosphorescent because they have a triplet state. And this is why you want actually a maximum of singlet oxygen generation and nearly no phosphorescence. Because, if you do have phosphorescence, you lose actually for singlet oxygen production. So it is a compromise to have.
And so one very interested question from David Agar. So can you link the compound to HSP inhibitors and greatly accumulate it in tumors to improve specificity? So could you link it to a protein?
Actually, we are currently trying not to work with this stuff, but we are currently actually working with antibodies, with peptide, and with other types of inhibitor to have a double effect indeed. This is really a strategy which is very, very interesting for certain types of tumors. So yes, you're absolutely right, David.
Okay, so thanks a lot. I think we should thank and upload all the speakers from this session. It was extremely interesting. And maybe we should take, we said we would take a picture. So maybe if you could all turn your cameras on. So the concluding remark is, I think, it was a very interesting meeting, even though it was very short, but we had a flavor of the diversity and the richness of the science that is being done both at QBI in UCSF, but also on the Montagne Sainte-Geneviève in Paris via the QLife Institute. And I think what this initiative is really specifically interesting is because first we observe the various scales of the living, which I think is super interesting, and a major issue in biology now is to reconcile scales. That's very tricky problem. And also, I think while there are many interactions between biophysicists and mathematical modelizer with biologists, there are less interaction, and I think we should really encourage this. And the last talk, I think, really showed that interaction with chemists and biologists. So I thought that was really interesting, and I learned a lot, at least for me. So thanks to all the speakers.
maybe I just chime in. Obviously, this was supposed to be in person here in San Francisco. We will have the next one in person in San Francisco next year, hopefully not be earlier in the year versus late in the year. And I thought the talks were fantastic. I really appreciated the participation from the virtual audience as well.
Yeah.
So, and our goal now is to announce another RFA soon so we can facilitate collaborations that'll hopefully have stemmed from these 2 great days of talks. So I'll just end there. I don't know, Bruno, did you have anything to say at the end?
I think we should, we should go on this collaboration. The talk were very interesting, and I'm looking forward to other collaboration that will arise from this meeting. So I think it was a very good experience. We should move on.
All right, thank you, everybody.
Do we take a picture or shall we go?
Oh yeah, was there a picture? Everybody's leaving.
Yes, a picture with the speakers.
Oh, okay. I took one screenshot if you want. Yeah.
Excellent, excellent. And if the others can join us, that would be great.
But Gilles is already gone.
Oh, we could
quickly.
All right, here we go. Here we go. All right, Carolina, on 3. 1, 2, 3. Excellent.
Okay, thanks everybody.
Thank you. Bye-bye.